Today we learn about the abscicic acid and have learned about four things. The first thing is the discovery of abscisic acid, biosynthesis, transportation, the metabolism of abscisic acid. Third is the main function, and the fourth the relationship with the other hormones.
In mid-1960s, three independent researchers reported three inhibitors. First, inhibitor B. Second, abscission 2. Third, dormin. Later, it was found out these three substances chemically were identical, and they were found as abscisic acid, or for short, ABA. It is growth inhibitors. ABA was isolated by two scientists, Liu and Kranz, from mature cotton fruits. Next, the occurrence of ABA. ABA is ubiquitous hormone in vascular plants. It is synthesized in almost cells that contains chloroplast or amyloplast. The structure of ABA determines its physiology activity. Naturally, is in seed form, and by convention, it is known as ABA. Any changes in structure results in loss in activities.
Now we come to the second part is the biosynthesis, metabolism, and transportation.
So, I will continue with biosynthesis, metabolisms, and transport of ABA. The first one is biosynthesis of the ABAs in chloroplast or other plastid. The pathway begins with IPP is mean isopentanins diphosphate with the 5C and use the finger page 23.2 in textbook. Surprisingly, in plants, ABA is synthesized not from the C15 secutterpenes, precursors phenexins diphosphate, but in the hydroxylase carotenoids called the zeaxanthin. Zeaxanthin is converted to the violacanthin by the enzyme zeaxanthin oxidase RES-EP. Violacanthin is then cleaved to form the compound called the xanthocene with the 15 carbon. Ungrowth inhibitors by the enzyme NCED. It means carotenoids dioxidase, and this is the first committed step in the ABA biosynthesis. Finally, xanthonins converts to the Abscicic aldehydes, and thence ABA mutants defective in the ABA synthesis have to prove to be very useful. The second one, ABA biosynthesis and concentrations can dramatically fluctuate in the different plant species during development in response to environmental stress caused by 5 effects. Biosynthesis regulates ABA concentrations in cytosol degradations, its compartment tutations, conjugations, transport. The third part with the transport, with the two ways of the ABA transportations through polymer and xylem. In polymer, more abundant with a radioactive ABA in the leaf and up to stem and down to roots. In xylem, from the roots to shoots. With the well-watered plants, ABA concentration greater than the water strength. The magnitude depends on the plant species. Also, ABAs can be transferred in their conjugate form, then hydrolysis in the leaves in the short sections by their hydrolase. So, among plant cells compartments, ABA distribution follows the onion strap concept. Same in textbook.
Now we come to the third part is the function. Abscisic has three functions. The first one is the dormancy, mean the sleeping of the seed. The second function is ABA being a stress hormone, closing the stomata and increasing the root-shoot ratio to maintain the living of the plant in a dry environment. The third function is the abscission. Now, let's come to the first function, dormancy.
Control of seed germination and growth is crucial to the survival of the next generation, and there are critical checkpoints at transition from dormancy to germination and from germination to growth. Seed dormancy increases the likelihood that seeds will germinate only when there are sufficient amounts of light, temperature, and moisture to the seedlings to survive. Two types of seed dormancy have been recognized, called imposed dormancy and embryo dormancy. For the first one, dormancy imposed on the embryo by seed coat and other enclosing tissue, such as endosperm, pericarp, or extra flower organs. The embryos of such seed will germinate readily in the presence of water and oxygen once the seed coat and other surrounding tissue have been either removed or damaged. The second type of seed dormancy is embryo dormancy, a dormancy that is intrinsic to the embryo and is not due to any influence of the seed coat or other surrounding tissue. Different types of seed dormancy also can be distinguished on the basis of the timing of dormancy onset rather than the cause of dormancy. Seeds that are released from the plant in the dormant state are said to exhibit primary dormancy. Seeds that are released from the plant in a non-dormant state but that become dormant if the conditions for germination are unfavorable exhibit secondary dormancy. What prevents seeds dispersed in autumn from germinating immediately, only to die in the winter? What mechanisms ensure that such seeds do not germinate until spring? For that matter, what prevents seeds from germinating in the dark, moist interior of the fruit? The answer to this question is ABA. The levels of ABA may increase 100-fold during seed maturation. The high levels of ABA in maturing seeds inhibit germination and induce the production of proteins that help the seeds withstand the extreme dehydration that accompanies maturation. Many types of dormant seeds germinate when ABA is removed or inactivated. The seeds of some desert plants break dormancy only when heavy rains wash ABA out of them. Other seeds require light or prolonged exposure to cold to inactivate ABA. Often, the ratio of ABA to gibberellins determine whether seeds remain dormant or germinate, and adding ABA to seeds that are primed to germinate makes them dormant again. Inactivate ABA or low levels of ABA can lead to early germination.
So, we come to the next function of abscisic acid is being a stress hormone. During the water stress-like drought condition, very dry and very arid environment, the abscisic level of the leaf can increase 50 times. In addition to closing stomata, ABA increase the hydraulic conductivity of the root and increase the root-shoot ratio at low water potential. As the soil dries, abscisic acid ascends from the root to the shoot, and the increase of pH of the xylem sap may allow more of the abscisic acid of the leaf to be translocated to the stomata via the transpiration stream. And when this increased level of abscisic acid travels into the gut cells, it can interfere the uptake of calcium, potassium K+ by the gut cells and simultaneously pump out K+ from the gut cell. This K+ lag make the osmotic pressure decrease and simultaneously make the stomata lose its turgidity. And of course, after that, the gut cells is partially closed. So how can the gut cell pump out K+ and inhibit the K+ pumped in. So we have this picture is the pathway. We have two pathways, the ROS, reactive oxygen pathway, and IP3, cyclic ADP ribo pathway. So the main principle you have to remember about these two processes is they simply increase the calcium 2 plus inside the two guard cells, and then this calcium 2 plus have inhibit the K plus in channel. And although the rise in intracellular calcium promotes the opening of chloride ion channel on the plasma membrane, so it causes the membrane to be depolarization. The plasma membrane proton pump is inhibited by the ABA-induced increase in the cytosolic calcium, because the calcium increase have also released the calcium from the vacuole and further depolarizing the membrane. The membrane depolarization activates K plus ion channel, and K plus and anion to be released across the plasma membrane are first released from vacuoles into the cytosol, so that K plus is just pumped out and not pumped in, so that it makes the osmotic process to decrease and stomatalysis turgidity and partially close. So the other process of coping with the stress that we want to talk about is increasing the root-shoot ratio of abscisic acid at low water potential. So abscisic acid has different effect on the growth of root and shoot. Officially saying, and the effects are strongly dependent on the water status of the plant, compares the growth of shoot and roots of a maize type, maize corn, under either abundant water condition, high water potential, or low water potential. They have compared two types of seedling. It's the normal ABA level seedling and ABA deficient, ABA lack seedling. When the water supply is high water potential, the shoot growth is greater in the normal ABA than in the ABA lack mutant. The reduced shoot growth in the ABA deficient mutant could be due in part to excessive water loss from the leaf. So this finding suggests that endogenous ABA promotes shoot growth in well-watered plants by suppressing ethylene production. When water is abundant, root growth is slightly greater in the normal type. When the water is limiting, i.e. at low water potential, the opposite occurs. Shoot growth is greater in the abscisic acid deficient mutant than in the normal type. Thus, the normal ABA acts as a signal to reduce shoot growth, but under the low water condition. However, the growth of the root is much higher in the normal type than in the lac type. So, to summarize this experiment, under dehydrating condition, when abscisic acid levels are very high, the normal hormone exerts a strong positive effect on root growth by suppressing ethylene production, and a slight negative effect on shoot growth. The overall effect is a dramatic increase in the root-shoot ratio at low water potential, which, along with the effect of abscisic acid on stomata closer, these two processes helps a plant to cope with the water stress and help it survive throughout the dry season.
Last but not least, the third function of abscisic acid is involving in the abscission process. So normally, in the abscission process, oxime and ethylene is usually considered as a couple of hormones that control the abscission. But actually, the abscisic acid is obviously a very important hormone in this abscission process, involving in the leaf and flower abscission, and it can increase the formation of ethylene or individual not dependent on ethylene to cause the abscission.
Okay, so my part is about some key relationship of ABA with other hormones. First, I will remind you about the domain C. The domain C is divided into two parts, non-dominant and dominant. In non-dominant, seed need only water to germinate, and in dominant, seed fail to germinate in presence of water, and instead it requires some additional treatment or condition. So in relationship of ABA with GA, both ABA and GA can regulate the seed dormancy. The ABA with major peak present in seed help suppress the vivipary in mid-embryogenesis. The ABA mutant reduce the seed dormancy, and the GA will help stimulate the plant to grow. Most plants in the peak of ABA production in the seed coincide with a decline in level of IAA and GA. IAA is the auxin, so GA deficient mutant cannot germinate with the absence of exogenous GA were mutantized. The seed produced by mutagenized plant can be regain their ability to germinate. However, they turned out to be ABA mutant because the dormancy had not been induced, so GA is no longer overcome. So as a result, the balance of ABA and GA in plant is very important, especially in regulating plant development. For example, GA and ABA are also important in bulb information. In bulb information is a result of balance of GA and ABA. The high content of GA will suppress the development of bulb root. The bulb and the high content of ABA will make the bulb easier to grow. So we can use some CCC as the resistance of GA to improve the bulb information. And next, in the relationship with of ABA with cytokinin, the senescence in plants related to the balance of cytokinin and ABA. The high content of ABA also makes the senescence, and the high content of cytokinin have reverse effects. So rejuvenation related to the cytokinin synthesis in root system, and senescence correlate with the plant reproductive system. About the relationship with ABA and ethylene, the abscission is related to the balance of auxin and ABA with the ethylene. In the abscess organ in plant, the content of auxin is very low, so ABA and ethylene is high.
Wow, I think that's mostly everything that we have talked to each other about. Thank you.
And the moments that you wandered far from me, I wanna feel you in my arms again. And you come to me on a summer breeze, keep me warm in your love, and then softly leave. And it's me you need to show how deep is your